Hello everyone, my name is Ahmed Mustafa. I'm an assistant professor of pathology and lab medicine at the University of Saskatchewan in Canada. I'm also serving as the clinical lab director of the histocompatibility and immunogenetics lab at St. Paul Hospital in Canada. Today I'm going to give us a short or a comprehensive introduction about unveiling the new histocompatibility and immunogenetics lab report about Eplet mismatch. So I'm going to start my presentation with some introduction about HLA and HLA antibodies, and then I will go into the difference between epitope matching and epitope analysis. And we will touch base about the new Eplet mismatch group report that our lab are using now. And then we will talk about the significance of this Eplet mismatch, and I'll wrap up my presentation with some of the case studies. So we will start with introduction to HLA and HLA antibodies. So the field of the major histocompatibility complex, or the HLA, started early in 1950s by a group of three different scientists. And here I will focus on Jean Dosset, who is a French scientist, and he got the Nobel Prize in 1980. So one day, Jean Dosset asked the assistant of his lab to bring him some blood, where he isolated the white blood cells from the puffy coat. At this time, they didn't know that the white blood cells express HLA, but I just added the HLA markers on the top of the white blood cells just for clarification. So he added one drop of this white blood cells on a slide, and then he asked the same assistant to bring him blood from people who has been multiple transfused with white blood cells. And he took one drop of the serum, which we know that the serum now contains a lot of antibodies, but at this time they didn't know. And he started to mix the serum with the white blood cells. And then he examined the reaction under the microscope. He noticed two different reactions. Sometimes the white blood cells get agglutinated by the serum, and sometimes the white blood cells does not get agglutinated. So he assumed there is some sort of biological material in the serum that sometimes interact with the white blood cells and sometimes not. But interestingly, what he also found that the patient own serum does not agglutinate the patient own white blood cells. And sometimes he noticed that by adding serum to the white blood cells, it results in massive agglutination, and sometimes it does not result in any agglutination. So he named this phenomena as MAC, or MAC, which represent the three letters or the three initials of his first three donors who donated some blood for this experiment. Later on, this MAC phenomena is known as the HLA-A2 because HLA-A2 is the most common antigen present in the Caucasian population. So what is HLA? So HLA is a human leukocyte antigen. Human because we know it comes from human. Leukocyte, as I mentioned before, all the initial experiment which was done by Jean Dosset and the other two scientists was done on white blood cells. And then antigen, because we know that HLA is expressed on the surface of white blood cells, and it can elicit an immune response in the form of antibodies. So now let's talk about HLA antibodies. So what is HLA antibodies? HLA antibodies are actually named according to the HLA antigen that they bind. And remember, there is a difference between antibodies and antigen. So for example, if this red antibody going to recognize the A1 antigen, then this is called the HLA-A1 antibody. If this antibody going to recognize the DR7 antibody, then this black antibody will be called the HLA DR7 antibody. And if this yellow antibody going to recognize the CW9, then this antibody is called HLA CW9 antibody, and so on. However, the idea of binding the antibody to the antigen is not as simple as I mentioned before. And here I want to clarify, what does the antibody actually see on the surface of the antigen. So here I put an example of the CW9 antigen. And if you look more carefully, you can see that this antigen has 6 different epitopes. For example, it could be, this is just like for schematic representation, but in real life, it has hundreds of epitopes. So if you look at the surface of the antibody, you can recognize that this antibody will match the yellow epitope. So in the reality, this antibody will only match to the yellow epitope present on the CW9 antigen. The same idea is the same idea of the lock and the key. So if you have multiple keys, only one key will be able to open this lock, and the other keys will not be able to open. And this is a very simple schematic diagram on how does the antibody recognize or bind to the antigen. So since we started to talk about epitopes, let's go into more details on some of the definition. The first definition is epitope, which is actually 2 words. Epi means upon and top means place. And epitope is a short set of amino acid sequence in the antigen that is capable of stimulating an immune response. The next definition here is the idiotype. And as you see in the figure, it is the polymorphic region of the heavy and the light chain of the antibody. The third definition is idiotopes, which is very clinically significant. It is the antigenic determinant found on the idiotype. These part of the antibody that are targeted by some of the therapeutic antibodies, like the IVIG that we commonly use. And this IVIG binds to the HLA antibody through the idiotopes. That is why it's called as anti-idiotope antibody. Lastly, the paratope, which is part of the antibody that recognize and bind to the epitope. So it's actually the lock part of the key. You can see the difference between the heavy and the light chain here. This distance is measured in angstrom, and this distance is approximately 15 angstrom radius. So I know that this slide looks to be a little bit complicated, but let's go slowly to understand this slide. This is a 3D crystallography structure of an HLA class 2. So what you see here in blue, this is the beta chain of the class 2. What you see in purple here, this is the alpha chain of the class 2. And what you see in brown here, this is the peptide binding groove of the HLA class 2 molecule. I found that this slide is very informative because it actually can show you the difference between polymorphic amino acids seen in yellow. And if we go a little bit bigger, you can see the eplet, which is shown in red. And if we go more bigger, you can see the epitope, which is shown in green. And you can notice here that the radius of the eplets is within 3 angstrom radius, and the radius of the epitope is between 15 angstrom radius. And if we go back to this slide, and you can measure the distance between the heavy chain and the light chain of the antibody, you can see that this distance is within the 15 angstrom radius, which actually match the epitope. So let's summarize what we learned so far. So Eplet is a short sequence of polymorphic amino acid residue. It could be linear or could be discontinuous sequence. It must be exposed on the cell surface because this is how the antibody will see. If it is buried inside the antigen, the antibody will not see it. And this is why I am stressing that the Eplet must be expressed on the cell surface. It must be non-self, because if it's a self epitope, antibody will not bind to a self epitope unless there is some sort of autoimmune disease. And it should occupy a radius of 3 angstrom radius. So before moving forward, let's explain an important phenomena that is shared between most of the antibodies. This phenomena is known as the cross-reactive group, or the Craig group of antibodies. And here I put some examples of 4 different antigens. What you see here, this is, for instance, this is HLA-A2, this is HLA-A23, this is an HLA-24, and HLA-68. And these are 4 different HLA antigens. What you see here, I just put a schematic diagram to represent, for simplicity, I just added that this antigen is formed of 4 different epitopes, and the same for the others, just for simplicity. So if we have an antibody that is going to bind to the HLA-A2 antigen, this antibody will not actually recognize the HLA-A2, but it will recognize a specific epitope present on the HLA-A2. If this epitope is shared between the 23 and the 24, then the same antibody will bind to the 23 and the 24. Let's take another example. If you have an antibody against this purple epitope of the HLA-A2, the same antibody will recognize that HLA-24. And the same scenario happen with the other epitope here, and this one here, and the rest, as you can see. Very important to know that there are some epitopes that are only very specific to certain antigen, and this is called the allele-specific antibody. And you can see an example of an allele-specific antibody is the yellow diamond epitope here, and the green circle present in the 23. So because they are not shared between any other antigen, then only one antibody will recognize this epitope. And this is what we call allele-specific antibody. So let's see why this is important in the clinical scenario. Here we have a donor that is willing to give a kidney to the recipient. If you look at the HLA typing of the donor is typed as A3, A2, B51, and B57. And if you look at the recipient is HLA typed as A3, A31, B51, and B8. So you can acknowledge that there are 2 antigen mismatch between the donor and the recipient. Now, if, for example, this patient develop some sort of virus and you have to reduce the immunosuppressive agent, now this patient has been already sensitized against the A2 and the B7. But in reality, when it forms antibody, this patient will form an antibody against the whole A2 cross-reactive group, and the whole B7 cross-reactive group. This will result in a CPRA, or a calculated panel reactive antibody, of 88%. This means that this patient will only likely to have 12% chance to find a suitable match in the future. Now let's switch gear and talk about epitope matching. So in this slide, I listed one patient who schematically have 4 different epitopes. And we have 3 donors that are willing to give kidney to this patient. So if we want to know which is the best match donor to this recipient, then the best thing is to compare the epitopes to the epitopes. So if we took the first example here, you can see that donor A has 4 mismatched epitope to the patient. If we look at donor B, you can see that they are matching on 1 epitope, then that means that they have 3 epitope mismatch. But if you look at donor C, you can see that donor C is matching at 3 epitopes, but only have 1 epitope mismatch. So in conclusion, this means that donor C will be the best fit for this patient. Why? Because if this patient is going to develop a sensitizing event against this donor C, this antibody will be only one antibody, because this is one antibody that recognizes this specific epitope. But if we use donor A, for example, and then the patient develops sensitizing event, then the patient will develop sensitizing event against the 4 epitopes. I hope this is clear now. So let's take another example. Here we have a donor that is typed as HLA-AB18. Because he is HLA-B18, then if we look at his HLA-B18 antigen, you can find that there are 11 different epitopes are present in different colors, as you can see. Now, if the patient or the recipient is typed as HLA-B7, then the patient does not have the same epitopes that the donor has. Because of that, the patient may develop some sort of antibodies against the donor. And because these epitopes are different, then the patient is most likely going to develop 11 different antibodies or 11 different sensitizing events. But we need to look at the whole picture of the patient. Now, if we look at the patient other typing, the patient is typed as HLA-B52 and HLA-A33. The combination of these 2 antigens, if you look at the epitopes, you can see that the patient is actually matching the donor typing. So in that, what does that mean? This means although there is 1 antigen mismatch, which is the B18, and the patient is B7, so it's 1 antigen mismatch, but there are no epitope mismatch. So this donor and recipient will have 0 mismatch of epitopes. So although they have 1 antigen mismatch, this patient is unlikely going to develop antibodies against the donor, because the epitopes now are considered self-epitope, and the patient will not develop antibodies against their own self-epitopes. So now let's discuss how is the Eplet named, or what's the Eplet nomenclature. Eplet is formed of a bunch of number and letters, as you can see in this example here. So let's understand what these number and letters are. If we look at the first epitope as an example, 80T, 82L, and 83R. The letters here represent the amino acid. So T stands for threonine, L stands for leucine, and R stands for arginine, if you still remember some of your biochemistry background. The number represents the location of this amino acid in the molecule. Since these numbers are not consecutive number, as you can see, it's 80, 82, and 83. There are missing number in between. So this is now considered non-consecutive numbers. You can know that this is some sort of what we call discontinuous eplet. For simplicity, it's usually written as 80 TLR. Let's take another example. The next example we have here is 81A, 82L, and 83R. Now you can see that the numbers here are consecutives, mean there are no missing number, 81, 82, 83. So this means that this eplet is in the form of a linear or composed of 3 linear amino acids, which is known as triplet. The next 2 eplets are an example of an eplet formed of 2 and 1 amino acid. So this is just like the software that we use in the lab that compare between the donor and the recipient typing. So for example, this program is called the Fusion software program. And this Fusion software program is connected to another online version called the HLA Matchmaker. And the HLA Matchmaker actually compare the typing of the patient to the typing of the donor and give you the number of mismatches. So if we look here at the first column, you can see that I listed the patient, which is P1 4 times. So it's exact typing listed 4 times. And in the next column here, you can see there are 4 different donors. Donor 1, donor 2, donor 3, and donor 4. So now basically I'm comparing patient 1 with 4 different donors to see which will be the best donor fit for this patient. The program will look at the patient typing and compare it to the donor, and will list you here all the eplet mismatch between the patient and the donor. And in the next column, you can see the number. So if you count these eplet here, you can see that the total number are 10 eplet mismatch between the patient and the donor. If you look also at donor 2, you can see that also donor 2 has 10 eplet mismatch. But if you look at donor 3, you can see that donor 3 has 7 eplet mismatch. So if we have the privilege to pick which is going to be the best donor for this patient, then it's definitely going to be donor 3, because donor 3 has the least number of eplet mismatch between the patient and the donor. So let's see how do we implement this eplet mismatch into our current report. So this is a regular report that we have in our lab. You can see here in the top panel, this is the patient typing, and we type all our patient by high resolution typing. And in the bottom panel here, you can see this is the typing of the unrelated donor. And in here, there is another row which says mismatch in the graft rejection. So this is basically we are comparing the patient typing to the donor typing at the antigen level, not at the allele level or not at the eplet level. It's only at the antigen level. So for example here, the patient is typed as A0101 and 2601. The donor is typed as 2402 and 6801. So this means these 2 antigens are different than the patient. So this get the score of 2. The same scenario happened with the B. And if we look here at the C, you can see at the antigen level they are matched, 07 with the 07 here, but they are mismatched at the other antigen, 03 and 12. So they got the score of 1. The interesting point here is, here this is the DRB53, which is the DR52 antigen. So both the donor and the recipient will have the same antigen, although they have different alleles, but they have the same antigen because both of them will be the DR52, so they will get a 0 mismatch. And here you can see it's going to be one mismatch, because if you compare the 0301 and the 0402 with the 1301 and the 1302, there is only one antigen mismatch, because the 13 is now considered homozygous, so it's only the 13 is considered one antigen mismatch between the patient and the donor. But let's see what does the Eplet will give us. So here, if we compare now using the HLA Fusion software and the HLA Matchmaker, it will give you the number of Eplet mismatch. So now, although these are two antigen mismatch, but there are actually 19 Eplet mismatch. Here is 16. Here is, although it's only one, it's now 10. This one I found it very interesting, because although you have one antigen mismatch between the patient and the donor, you can see they have zero eplet mismatch. This means that there are some of the epitopes shared between the patient and the donor at the eplet level. So they got zero eplet mismatch. So although if we only look at this data, we will think that the patient can develop one antibodies against the donor, but if we look at the eplet mismatch, you can acknowledge that there are zero eplet mismatch. This means that the patient will not develop antibodies against the DR beta 1 of the donor. The other thing which I found interesting here, although that we think because they are sharing the same antigen, which is the DR52, which is 0, but they are actually 6 eplet mismatch, so they can actually develop allele-specific antibodies against the donor. So the previous slide was only a slide to show you the difference between eplet mismatch and antigen mismatch. But our report for eplet mismatch group report is, this is what you see on the screen now. We call it the eplet mismatch group report, and also we put the patient typing on the top and the donor typing in the bottom, and then we do the comparison between the patient and the typing, but we don't put the antigen mismatch, just the eplet mismatch. And we put here our cutoff was based on a published paper by Chris Wibble group from Manitoba, and this group only studied the class 2. So they studied the HLA DQ alpha, DQ beta, and the DR beta 1. And they stratified the patient into 3 different levels. If you have 0 eplet mismatch, you are in the low risk. If you have between 1 and 10 eplet mismatch, you are in intermediate risk. If you have more than 11 eplet mismatch, you are at a high risk. And now, based on this publication, although we put the eplet mismatch of the A, B, and C, we don't have any published data so far about the risk assessment of the class 1. But we have risk assessment on the class 2 for eplet mismatch. Our lab is now interested in doing some studies about class 1, and we are currently doing some sort of research project to determine the risk assessment associated with class 1 as well. But based on this publication, you can see that this patient has a 0 eplet mismatch on the DR beta 1 and a 0 eplet mismatch in the DQ beta. So this is what we put on the final comment, that eplet mismatch are 0. So this means these donor and recipient are considered low risk for transplantation. And here is another group report that comes from our lab. But in that case, if you look at the number of eplet mismatch, you can see that the eplet mismatch for the DR is 12 eplet mismatch, and for the DQ beta is 23 mismatch. So in our comment here, you can see that there are 12 eplet mismatch and 23 eplet mismatch at the DQ. So now these pairs are considered high risk. And this is a snapshot of the paper published by the Chris Wibble group from Manitoba. about the comparing the antigen mismatch to the eplet mismatch. And this is the reference that we used in our report. So let's look at this paper more carefully, or at least at these slides. So you can see here they used a cohort of 600 patient and donor pairs. And this was a retrospective study. They divided these patients into 4 groups. Group 1 has one antigen mismatch, and be careful on what you hear. I'm calling antigen mismatch, not eplet mismatch. Group 2 has 2 DR mismatch. Group 3 has 1 DQ mismatch, and Group 4 has 2 DQ mismatch. And then they counted the number of eplet mismatch in each group. Each dot here, what you see is one patient. So each dot is one patient. So for example, if you look at this patient here, you can see that this patient has few eplet mismatch. But if you look at the top patient here, they have 30 eplet mismatch. And the same scenario you can see in group 2, 3, and 4. So it's very difficult to say that if you have one antigen mismatch, you will have less number of eplet mismatch. And I will show you an example shortly. So for example here, if we look at these 2 patients, although this patient has one antigen DR mismatch, and this patient has 2 antigen DR mismatch, but if you look at the number of eplet mismatch, this patient has almost above 30 eplet mismatch, but this patient has less than 10 eplet mismatch. So if you are comparing these 2 donors together, and the patient is asking you, should I, or you are asking yourself, should I proceed with the donor who has one antigen mismatch, or the donor that has 2 antigen mismatch? The answer to this question, you need to do eplet mismatch first, because if you pick based on the old theory that one antigen mismatch is better than the 2 antigen mismatch, then you are actually missing a lot of data about the eplet mismatch. So in that case, this means that this patient is better than this patient, or have a low risk than this patient, because the number of eplet mismatch are less. This means there are less antibodies are going to form. So this figure has been taken from a Chris Whipple group from the published paper. And this is actually a de novo donor-specific free survival curve. And in the lab, we usually do this curve all the time to monitor the production of donor-specific antibody. But based on Chris Whipple finding, you can see that he stratified the patient into 3 different groups. Group 1, which is in green here, is the group that has 0 mismatch. And also, this is the group that did not develop any donor-specific antibody. Group 2, which is the intermediate level, who has between 1 to 11 eplet mismatch, and they develop some sort of donor-specific antibody. And the worst group here was the group who has 12 to up to 41 eplets mismatch, and these are the group that developed more donor-specific antibodies. So I know a lot of audience here will start arguing with me that eplet mismatch is not the sole predictor of the graft survival or the formation of donor-specific antibody. And I have no issue on that. And based on these, Chris Whipple group did another study where they looked at adherence in combination with the eplet mismatch. So they used a group of 195 patients, and they give them this electronic tool bottle cap. So every time the patient open the medication and take their medication, it counts on this electronic cap. After a couple months, they collected this cap, and then they started to record the adherence of these patients. And after 2 months, they were able to group 44 patients, which represent 22.6% of the total patient, who had less adherence to the medication. And when I say less adherence, this means they have 7% or more. In the first 2 months post-transplant, they lost some of their medication. And they monitored them over the period of 12 years. And you can see here that patients who were non-adherent at the beginning, they tend to have more donor-specific antibody. Patients who were adherent here had less donor-specific antibody formation. And why this is important, and this is what we will look at in the next slides. So the next aim that they did in this study, they stratified these two previous groups into two more subgroups. So now we have adherent and non-adherent. And then the adherent, we have patients who had less eplet mismatch, less than 10, adherent with more than 10 eplet mismatch, and then we have non-adherent with less than 10 eplet mismatch, and non-adherent with more than 10 eplet mismatch. And you can acknowledge that the worst group here was the non-adherent who has more than 10 eplet mismatch. So both adherence and the number of eplet mismatch can predict the graft survival. So if you have a patient who has very high number of eplet mismatch, you have to be very cautious in talking to them about the risk of developing donor-specific antibody if they are not very adherent to their medication. The CTOT9 study, this was a common study, and it was a randomized multi-center international study which was aiming to recruit 150 patients. However, it was stopped early. The idea of the study was to find the lowest risk possible patient that they could pull back on tacrolimus withdrawal on this group of patients. These low-risk patients are defined who has a living donor, no donor-specific antibody at transplantation, has a PRA of less than 30%, and they were giving the most up-to-date medication, which is thymoglobin, tacrolimus, MMF, and prednisone. They were followed closely for 6 months to ensure that they have no signs of rejection. They had a 6-month protocol biopsy to confirm there is no subclinical rejection, which was also confirmed by repeated single antigen bead assay at 6 months to prove they have no de novo DSA. Only at the time they can be randomized into one of the two groups, either withdrawal of tacrolimus or stay on the drug. But what happened after 21 patients has been randomized into the ratio of 2:1, so it was 14 on tacrolimus, 14 with no tacrolimus and 7 with tacrolimus. The study was stopped due to high incidence of the de novo donor-specific antibody-mediated rejection and the T-cell-mediated rejection. So they concluded that quiescence or doing well for a time does not actually equate with low risk. But what we will see interesting, as you can see, how if this study was going to be completed in the future. So here is another evidence of the importance of eplet mismatch. This is the CT9 study that I talked about, where they tried to withdraw tacrolimus from the low-risk patient, but the trial was stopped by safety board after 14 patients have diagnosed with rejection. These 14 patients, 5 developed DQ antibody. All of these patients have DQ eplet mismatch above the cutoff threshold. And although it was only 14 patients, this was statistically significant. Even what I found impressive is a few weeks later after the study was locked, the patient circled in red actually went and developed donor-specific antibody. and the patient was off drug for only 2 days before they developed T-cell mediated rejection. So if we remove these 2 patients into the group that developed immunological event, you can see that the method of risk stratification was actually impressive in stratifying this patient even with very low number of patients that we're dealing with. This suggests even if the study is to be repeated, Eplet mismatch must be used as one of the criteria for the low-risk group. So now let's switch gear and talk about Eplet analysis. But before we talk about Eplet analysis, I need to talk about the principle of the Luminix assay. And this is the assay that we use in the lab to quantify or to measure the antibodies strengths in the serum of the patient. So what is the principle of this Luminix assay? First, you take some serum from the patient, which we know that it contains the antibody, and we combine this serum with beads, and this is beads that we buy in the form of kit from the vendors, and each of these beads are coated with a specific antigen. So what happens when the antibody binds to the beads, and if it recognizes the same antigen, then these antibodies will bind to the specific beads. In order to detect which bead bind to the antibody, you add another secondary antibody that will bind to the primary antibody attached to the bead. And then you measure the fluorescence intensity using the Luminix 200 machine. So let's talk a little bit about these beads in this figure here. So for example, here you have 100 beads present in one aliquot. Each bead, as you can see, has a different color, and each bead has a different number. So here I took bead number 13, bead number 25, bead 67, and bead 88 out just to see the difference in the color that they have, and also to highlight that each bead of these has a specific antigen attached to it. So for example, bead number 13, as you can see on the right side here, has the antigen A1. Bead 25, which has a different color than bead 13, it might look similar, but trust me, it's a little bit different. It has the antigen A2. Bead 67 has the B7 antibody or antigen, and B88 has the HLA B8 antigen. So in the next figure, I will show you how this bead interact with the antibody from the serum. So for example, this is one bead here, and this bead has one of the HLA class 1 antigen. So when you add your antibody, when you add your serum to this bead, if the serum contains some of the antibodies specific to this bead, it will bind to the antigen present on this bead. And then you add your secondary antibody, because this antibody bind to the primary antibody that bind to the bead, and then this secondary antibody has a fluorescence tag in it. So when laser 1 hit the bead, from the different color of this bead, it will recognize which bead it is. And then when laser 2 hit the bead, it will recognize the strength of the antibody, because it's going to detect the fluorescence intensity present on the secondary antibody. So there are 2 different kinds of beads, or at least 2 different kinds. There are maybe 3 or more different kinds. But what I'm going to explain here are the 2 different kinds, which is the screening bead and the single antigen bead. So the screening bead mean that each bead contains different antigens on the surface of this bead. For example, here you can see this bead is specific for class 2, and you can see there are different colors of antigen attached to the surface of the beads. And this is called screening because it's not specific to a certain antigen of the class 2. The same here, you can see another bead which is specific for class 1. So all the different colors on the surface of these beads are different class 1 antigen. So basically, if you have an antibody that binds to this bead, you will not know which specific bead or which specific antigen is reactive on these beads. Like for example, here we can see in the class 1 bead, there is an antibody that recognizes the yellow color. So what we should do next is we should do something called single antigen bead. So the single antigen bead here, the difference between the single antigen and the screening is that each bead has only one color or one antigen attached to it. Like for example, this could be DQ1, this could be DR1, this could be A28, and if you notice here that this A28 is the same antigen that was present in the class 1, which is yellow. And this is why when the antibody bind to class 1, when we did the single antigen bead, you can see that the same antibody recognized the A28 antigen. And in that case, this case will be A28, or this patient has A28 antibody. Or it could be, for example, as the B60. So here is an example of the raw data that you can see that comes out of the Luminix, which is the machine that I was explaining before that analyzed the antibodies. What you see here in the x-axis are the beads or the number of beads binding to the antigen. As you remember, I mentioned before, there are 100 beads and each bead has a different color and each bead has a different number. From the color of the bead, when the laser hit the bead, it will tell you which, based on this color, it will tell you which antigen is bound, because the software is already programmed based on the color of these beads, which antigen is bound to it. And what you see here on the y-axis is the mean fluorescence intensity. So when laser 2 hit the antibody that is tagged by the fluorescence, it will flash up and will let you know that if the antibody is already bound to this bead or not, if you got a specific number. What you see here in the bottom is the corresponding antigen that is bound to the specific bead. Let's take an example. So if you look here at bead 63, which is the first bead here, you can see that bead 63 recognized the B46 antigen. If you took bead 77 on the top here, you can see that bead 77 recognize bead HLA B73 antigen, and the beads in the middle here recognize the CW antigen. So some of the labs use the cutoff of 1000 MFI. So anything below 1000 MFI is considered negative. Anything above 1000 MFI is considered positive. So if we are the lab that are using this cutoff as a 1000 MFI, so all these antigens will be considered as positive because it's above the cutoff. But there is some other labs that use the cutoff of 2000 MFI, and in that case, half of these antigens only will be positive. But let's talk about how we became more consistent in our lab in using the term of cutoff and identify which bead is positive and which bead is negative. But let's go to the first case study that I have here, and this will give you a better idea about how we call antibodies in my lab. So this is case number 1, which is Epitope explain the reactivity after a failed transplant. So on the left side here, you can see the patient typing. So the patient is A24, A29, B8, and CW17. And what you see on the bottom here, that this patient has got a transplant, but this transplant is failed. So now, if you look, you can see that the patient has developed antibody against the B46 because this is a mismatched antibody. But the patient did not develop anything about the B13 because you can see here the B13 antibodies is negative on the graph. The interesting part here that the patient and the donor was completely matched at the CW17. So how did all these CW antibodies that we see with very high MFI has been developed? So this can be explained by epitope analysis. And are these real antibodies or these are false antibodies? So let's see how can we explain it in the next slide. So here, how can we explain the reactivity of the CW, although both the patient and the donor were matched at the CW antigen? So we use the fusion software that I explained before, and we put the patient typing on the first column, the donor typing on the second column, and ask the software to pick up the different mismatches between the patient and the donor. And here you can see there are 6 different HLA mismatch, and there are 14 different HLA-AB mismatch. But what I found interesting is if you look at this specific eplet, which is the 76 VRN epitope, you can see that the 76 VRN epitope, because this was a mismatch between the donor and recipient, and as you still remember that antibodies are not formed against antigen, antibodies are formed against epitope. So because now this 76 VRN is considered a mismatched epitope, so we have an antibody that has been sensitized against this epitope, and now this antibody can cross-react with all these antibodies that you see. So one antibody that recognizes this specific 76 VRN epitope can cross-react and can explain the B46, the B73, and all these CW antibodies that you see in the patient serum. So even if we are using a cutoff of 2000, we will miss an important aspect of the story because it's actually real antibodies and you cannot hide them. It's formed because of the cross-reactivity of the 76 VRN, which was formed because the patient got sensitized against this specific epitope. Now, here is case number 2. And the question of this case, is it the DQ beta or the DQ alpha antibody that are giving this high MFI that you see on the y-axis? So this high MFI, could be the DQ alpha or the DQ beta. So let's remind you of the structure of the class 2 antigen. You can see the class 2 is formed of a DQ alpha and the DQ beta. And when they synthesize this bead, they cannot remove the DQ alpha from the DQ beta. So the same bead will have both the DQ alpha and the DQ beta. And you can see it here in this the figure here. You can see, for example, bead number 48. It is actually against the DQ5 beta, but also it recognizes the DQ alpha 0101. If you go to the next bead, which is bead 49, it also recognizes the DQ beta 5, but also it recognizes the DQ alpha 0102. So the question here, when you have something or some sort of reactivity like this, is that coming from the DQ beta or it's coming from the DQ alpha? And the interesting here is some of the DQ alpha is on the right and some on the left. Like some of the DQ beta is on the right, like here. All the DQ6 here are on the right side. So when you have DQ6 on the right side, this means they are negative because they have below 1000 MFI. But some of the DQ beta also is present on the left side, and this is the side that have high MFI. So is that actually going to be a DQ6, or now we consider this negative? Because some, in order to call them positive, all your beads should be positive. You cannot get half and half. But the other thing here also, if you look at the DQ alpha, you can see some of the DQ alpha is on the left side. This means they are positive, but also some of the DQ alpha 1 is on the right side, which means they could be negative. So should we call DQ alpha? Should we call the DQ6? And this is the main question we usually ask. So let's see how Eplet analysis can explain this to us when we call this specific reactivity of antibodies. So when we did epitope analysis, we found that epitope 75V can identify all these antigen on the DQ beta 5, 4, and then all the 2s except the 2, 6 here. But when we found another epitope called the 85VY, the 85VY is an allele-specific antigen or allele-specific epitope that only detects specific alleles in the DQ beta 6, but does not detect the other alleles. And you can see here all these other 6 alleles are negative, because only the 85VY is an allele specific, so it detects specific alleles of the DQ6. So when we click on to see the allele specific, you can see that the DQ6 is formed of different alleles. So these one which are positive are 0609, 0604, but the one which was negative are the 0603, the 0602, and the 0601. So this means these are allele specific antibodies. And if we are not going to use the Eplet analysis, we will not ever know that this exists, and this is actually a positive reaction. Otherwise, if we just use the DQ6 without the epitope, and since part of this DQ6 are negative, we will call them all negative. But because we use the Eplet analysis, we were able to confirm that these two are allele specific antigens, and we call them in our patient report. Another example here, and it's the same question, but it's a different case. Is it the DQ beta or the DQ alpha? So let's see how did the epitope analysis solve this issue. So you can see again here there is a bunch of DQ alpha and DQ beta positive. But which one is positive? Is it the DQ alpha alone, or it's the DQ beta alone, or it's a combination of both of them? So let's see what did the epitope analysis did for us. So when we did epitope analysis, the whole reactivity of the DQ alpha could be explained by epitope 40GR, and there was no clear reactivity explain the DQ beta. So in that case, this patient will have a DQ alpha antibodies and not a DQ beta antibodies, because there is no epitope can explain the reactivity of the DQ beta. So now we can conclude that in that case that the specificity that we see here, or the high MFIs, are coming from the DQ alpha, so it's coming from this side molecule here. And another interesting case here, which is the same question, because this is the most question that we get in the lab, and epitope analysis actually solve it for us. So here again, is it the DQ alpha or it's the DQ beta? So when we do epitope analysis, epitope 52PQ can explain the entire DQ6 reactivity, and epitope 18F can explain the entire DQ alpha reactivity. So in that case, it will be both the DQ alpha and the DQ beta are responsible for this reactivity. And so when we put it into the patient report, we say that this patient has the DQ beta 6 and 5 antibody, but also this patient has also the DQ alpha antibodies as well. So let's move on with case number 5, and it's again the same question, but it will have a different answer. Again, when we did epitope analysis, you can see that the entire DQ beta, which has the 5, 6, 4, 8, 9, all these are explained by just one single epitope. So it looks like this patient was sensitized against one epitope, which is the 45GV epitope. And because this epitope is highly immunogenic, they develop cross-reactivity against the entire DQ beta antigen. But when you look at the other epitope here, you can see that DQ alpha is partially explained by 52SQ. So that means that this patient has antibodies against the entire DQ beta, but only have antibodies against partial DQ alpha. So when we only call partial DQ alpha antibodies in this case. So this case, actually, I found it very interesting, because if our lab is using the cut of 1000 MFI, we cannot name any of these antibodies. We can even say that these antibodies are all false, because you can see here some of the DQ are on the left side, and some of the DQ are in the right side. Like, for example, 8, you have DQ8, and then you have DQ8 which is negative, and then another DQ8 which is negative here. And the same for DQ7. Some are positive and some are negative. So, and as I mentioned, you cannot call some are positive and some are negative unless they are allele specific. But when we did epitope analysis, you can see that this positive one and the negative one are explained by one epitope, which is the 55P. The interesting part here, which I'm going to explain in a different case, this is a well-known phenomena called the peanut butter spread, and just keep tuned, you will learn about it in the next few slides. But the idea here is sometimes you have limited amount of fluorescence or secondary antibody. When you have antibodies are spreading all over the beads and you have limited amount of secondary antibody, you sometimes get lower signals. So what you see here on the right side, this is just like lower signals of the antibody. This doesn't mean that they are absent, but they are just like lower signal because it's the limitation of the assay, because we use a certain amount of secondary antibody. Maybe if you add more antibody, you can get this excited more, but you have to be consistent between all your assay. So the take-home message of this case, if you use the cutoff of 1000, you are actually losing the entire reactivity here. But if you're using the epitope analysis, you can confirm that this is actually a real reaction. So the conclusion of the previous cases are: epitope analysis can explain allele-specific antibodies, as I mentioned. It can explain the partial DQ alpha reactivity, the partial DQ beta reactivity, or the combined DQ alpha, DQ beta reactivity. And if we move now to case number 7, you can see that on the y-axis here, that all the reactivity is below 1000 MFI. So should we call this as antibodies, or we just say they are negative? This is one of the difficult cases, because this, although they are negative based on the MFI, but they are clustered, all are clustered and can be explained by one specific epitope, which is the 65QIA. So there is a high chance that this patient received a blood transfusion maybe 10 years ago, and then his antibodies subsides, but once he got immunized again, it will flare up. So something like this, we need to get more cautious, and we might call them negative, we might call them positive, but at the end result that we have to be cautious that there is some sort of sensitization happening to this patient here. Maybe we can do some reflex testing like surrogate cross-match, or we can do other testing as well to confirm if this is real or not real antibodies. But in most of the cases, this is real because they are clustered. They are explained by one single epitope. So before moving into the case number 8, I just want to highlight an important feature that I need to discuss about epitopes, which is the denatured epitope. Sometimes when intact HLA molecules are isolated from the cell during the manufacture of the single antigen bead, they become denatured. Denature means that they will expose certain areas of the molecule which was not exposed before. As you can see in the 3D structure of the class 1 on the left side, it's an intact molecule because the peptide in brown is covering the molecule. But when it becomes denatured and lose the peptide, some of the hidden eplet become exposed as isoleucine seen in position 73 in yellow. When you perform single antigen bead assay, and if there is a non-specific antibody in the patient's serum that recognizes this denatured antigen, it will react and bound to this denatured antigen tenfold higher and result in very high MFI. This is usually referred as a hot bead in the single antigen bead assay, and we usually see this in a daily basis. These are considered false positive and accounts for some of the limitation of the single antigen bead assay. So case number 8 is actually an example of the denatured antigen that I was explaining in the previous slide, because here when we did Eplet analysis, there is no epitope that can explain all this reactivity. You can see there is some beads are 3000 MFI, some are 500, but you can see all these entire reactivity of antibody are not explained by epitope analysis. So in order to confirm that this is a non-specific reaction, we have to do another confirmation or another reflex testing. One of the tests that we usually do, instead of running a single antigen bead, we run the screening test, because the screening, the antibodies are more in the nature form. It's not denatured. So even this are non-specific, but they can tell you if there is anything positive or not. You can see that, like for example, B12, it has multiple antigen attached to it, like A2, 24, 54, 67, and so on, and all the beads are like that. But you can see clearly that none of these beads are interacting with the antibody present in the serum. So there is a high chance that the other reactivity was due to a denatured antigen present in this serum. So in conclusion of this case, I can conclude that this case was due to a denatured antigen. So if I concluded case number 8 and case number 9, mean fluorescence intensity cannot be used to determine the cutoff. You have to look for the patient reactivity. Eplet analysis can confirm the presence of denatured epitope, and another assay sometimes is needed to confirm reactivity, like the reflex testing that I was talking about. Another example of epitope spreading, if you have your single antigen bead cutoff set up as 1000, you will report this case as negative because none of the antigen are above the 1000 MFI cutoff. However, epitope ATI can explain the entire reactivity. It looks like a complete BW4 reactivity. And this is explained by epitope ATI. This situation has been explained as the peanut butter spread. So what is the peanut butter spread effect? Imagine you put a scoop of peanut butter on a piece of bread. Now picture that peanut butter spread across the whole loaf of bread. When you eat it, there is a little flavor on each piece. There is not less peanut butter or less antibody, it's just the spread out. I don't know if it will cause a positive physical cross-match. It could be a negative, but I predict it will be at least weakly positive. So why we think epitope analysis and epitope matching are the future? Because epitope matching and analysis are useful tools for all histocompatibility and immunogenetics lab. Of course, there are still more work need to be done, and the data is still evolving, but I think it's a good way in the right direction. And at the end here, I will stop my presentation, and I would like to thank all the audience for listening, and I would like to thank all my staff for their hard working at St. Paul Hospital at the University of Saskatchewan in Canada, and I will be have my contact information if you need it for future questions. Thank you.